I am going to talk about genes. Every cell in our body contains a copy of our genome, over 2,000 genes, 3 billion letters of DNA. DNA consists in two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and C pairs with C. Our genes shape who we are as individuals and as species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISP method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of the virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading viruses. These two RNA forms a complex with a protein called Cas9. Cas9 is a nucleus, a type of enzyme that can code DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 codes the target DNA, disabling the virus. Over the past few years, researchers studying the system realized that it could be engineered to code not just viral DNA. Genes also have profound effects on health, and thanks to advances in DNA sequence, researchers have identified thousands of genes that affect our risk of disease. To cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the When this happens, the cells try to repair the cut, but the repair process is error-prone, and ending to mutation that can disable that gene, allowing research. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method. These mutations are random, but sometimes researchers need to be more precise. For example, by replacing a mutant generic and healthy copy. This can be done by adding another piece of DNA that carries that desired sequence. Once the CRISPR system made a cut, this DNA template can pair up with the cut ends. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but any genes action together. These methods are being improved rapidly and will have many applications in basic research, in drugs development, in agriculture, and perhaps eventually for treating human patients with genetic disease. Thank you.